Hi, we are here for the Dig into Dyer Google Hangout with Marc Epstein. Marc is an entomologist and a research associate here at the National Museum of Natural History, and he's just recently written the book Moths, Myths, and Mosquitoes, The Eccentric Life of Harrison Dyar, Jr. And we have been we've been working in the libraries all week to encourage people to transcribe Dyer's field books, like this one here, which we'll see a little bit more of later. And Marc is going to help us give us some context to Dyar about his work and what he did. He also has a really fascinating personal life that we'll talk about too. So I'm Erin Rushing. I'm the outreach librarian for Smithsonian Libraries. As I mentioned, Marc is an entomologist and author. And so far, we have had, in the Dig into Dyer transcription campaign, we've had 5 of his blue books go up, and I believe about 80 volunteers have helped us transcribe this tricky handwriting here, and we appreciate all of your work. So if you are tuned in live and have any questions, please let us know. If you have any questions after the broadcast, you can tweet us at SI Libraries using the hashtag #DigIntoDyer. And you can also use the little Q&A box here on the side to let us know if you have any questions. We'll try to get to those through the broadcast too. So Marc, why don't you give us a little summary of what Dyar studied and why it's important today here at the Smithsonian.
Well, Dyar came here in 1897 at the request of Leland Ossian Howard, who was the lead curator and also the head of the Division of Entomology of the USDA. Like his predecessor, Charles Valentine Riley, he held both positions. The head curator was an honorary position, paid with the USDA as the U.S. entomologist. But one of the reasons why he wanted Dyar here is Dyar was really quite the expert on caterpillars and on moths, which sort of served both purposes, because the USDA was concerned about caterpillars being the stage that, you know, causes the damage, and then the collections, of course, did consist of both caterpillars and alcohol and dried caterpillars by a technique called blowing caterpillars, which we'll show you a little bit about later. But he needed someone that just knew moths, being a bigger group, part of Lepidoptera, which also includes butterflies. Butterflies really are just a type of moth, if you want to really look at it that way. A lineage that just split off at some point. There are other day-flying moths that are pretty just like butterflies, and all have members of each group that cause economic damage. Anyway, Dyar grew up in Rhinebeck, New York, collecting and rearing caterpillars, and at a fairly young age began to communicate with experts. At least by the time he was 16, the state entomologist of New York got advice for Dyar. Dyar would find sometimes that the moths were incorrectly identified because he knew that the caterpillars were different. So,
and this was at age of, in his late teens, he was?
Yeah, like by 19.
Wow.
But as early as 16 in the original blue book, we have Dyar's notes, and you can see
how tiny, tiny
little thing. And of course, we call all of the books blue books, but the only reason is because the original, the diary that he took from his mother, or was given to him by his mother, which she never wrote any diary things in it, just started an expense account information, but switched to other books. So
this was where I think this is so fascinating. So this was his first blue book that he wrote his observations in, and it was originally, it was not for field work by scientists. It was a home accounting book or home economy book that he took from his mom and used several years later, and crossed out the dates of 1876 and put in 1882 instead.
Correct.
And it's just so adorable. and was not what its original intention was, but that's
fascinating. And he started doing that in the middle of the book in June 1882, and then wrote through the field season, and then later on filled in the rest of the book with other notes that have later Blue Book numbers. And I will say something about Blue Book numbers now. They started out as a number that represented a particular species and its caterpillar, but not long after the 1880s, it began to just be an individual entry. So
that, for those of folks in our audience who are transcription center volunteers, and they may have seen the Blue Book numbers that are in the fours, 401 and 414 is one of the first Blue Books we have in the transcription center right now for the campaign. That number doesn't reflect the sequential order of the books, but it refers to the caterpillars that are described in.
Well, it becomes more sequential as time goes on, certainly. And actually that becomes a useful thing to look at. But also sometimes when it isn't, as he would, if he didn't fill everything in or found the same caterpillar later, sometimes he would in fact use the same number and go back and write and fill in the gaps, what was left. Other times he would write that same, usually though he would change numbers if he put it into a different Blue Book later on. So it became more or less sequential, though if he thought it was a slight variant or different species, he might use the number like 414A, 414B, because you have to remember that, you know, the entries in these books kind of came from different sources of the Blue Books. Some were, he'd find the moth and it would lay eggs, find the female, get the eggs, and then raise the larvae from eggs. So in those cases, he would often know what he was raising. In other cases, he never identifies them. He might put a skull and crossbones at the end because the caterpillar has died. Oh. So yeah, so there's a mix and match. So the plant that the caterpillar is feeding on is more important in the cases where he's found the caterpillar, because that's a true food plant for that caterpillar in nature. So that's actually useful scientific information. Whereas with his eggs, sometimes he knew already from previous experience, but oftentimes it was just a guess that he would use a common, you know, tree leaf from around there, and if it worked, he just used it.
He just used it. So would you like to sort of give us a tour of his field books and show us what some of these, I know some of our transcription volunteers have noticed the blue squiggles and the X outs, and would you like to, well, take the webcam off for a second and give you guys a closer look at his field books, and then we'll talk about the relationship between the field books and the specimens, which now you're seeing we have a sneak peek up there of some of Dyer's own specimens. So would you like to show us some of the squiggles and interesting annotations in the field book?
Okay, so kind of like when you go to Costco and you're checking out, well, sometimes they put a little check mark on your bill. Other times they do the little squiggle. And of course, since I've seen so many of these, they think I'm crazy, but I say, could you do a squiggle for me?
My kids prefer the smiley face, but.
Oh, they do? So here you can see some, and they kind of change over time, and there's the blue, there's the, you know, red check mark, and sometimes he'll, so what that means is that he had transcribed these notebooks into publication. So that's important to you as transcribers, in that one of the best ways to learn, as somebody wrote, they're just learning how he wrote the word brown, you start to recognize after a while. But another way to do it is if you can find where he has actually published the articles, and BHL, the Biodiversity Heritage Library, is a wonderful tool. The only thing is you would have to know, you know, more precisely what species we're talking about, and sort of have a good idea. Sometimes he actually says in here when and where it was published, but oftentimes he doesn't. So by getting the actual literature of the published, he didn't change what he wrote in his notebooks all that much. So it really helps with transcription, seeing the printed version.
And I think this is interesting. And one thing we haven't mentioned yet is Dyers Law. And one of Dyer's legacy to the field of entomology is Dyers Law. Could you explain that a little bit and what these little notes are?
Yeah. So let's see, where's it? Okay, we can take this one here. So on the top of this page, and this is entry 571, and he's got a little question, Artean question mark, and it's on Coccoloba floridana. So this is when he was at Lake Worth, Florida, which is, I believe, near Palm Beach, December 27th, 1895. So this notebook is a little bit after the ones that you've been looking at for the transcription challenge. Anyway, these numbers on the top, notice how it says calc, C-A-L-C period. It's got this series of numbers. And then down below, actually, let's find a better one. It doesn't have the actual. Okay, here we go. Here's another one. Also, is this one from Florida? Anyway. Okay, so he has calculations, and then at the end he has ratio, 0.7. And then below, he says found. So what Dyer did is that when he was raising, and this one was an egg laid by a female, two female things, the species here, found on whatever. So he got eggs. So what he did is that through this whole description, not only does he have a little drawing of the first stage caterpillar that hatches out, and sometimes these have never been, even if he says he's published it, that doesn't necessarily mean these little pictures he does, and he's a very good artist. They're not always published. But anyway, what he does is he measures the width of the head capsule, and that's the hardest part of what we call sclerotized. So it's the part of the skin of the caterpillar that is hard. So in other words, it doesn't change much within a stage. So caterpillars, remember, are insects too. They're kind of soft-bodied, but in order to grow, they have to shed their skin, and oftentimes at least 4 times, and maybe as many as 10 or 11.
Really? I didn't know that.
Yeah. So they have to shed, and each one of those molts or sheds, which we call instars, in the literature, Dyer kind of got frustrated because he would have raised a caterpillar that had 5, instars and molted, 6 times or whatever. But then somebody else might have had only 4. So he thought, well, they can't both be right. So how would I develop a method to tell how many there were, or a way to predict if, say, I find a caterpillar, can I predict what stage it is? So what he did was he took all his found measurements. He took 1.7 in this case over 2.4 millimeters, and you get a ratio. I'm not sure if that comes out 0.7, but it probably does somewhere close. But what's interesting is that he looked at the ratios across to see if it followed what we call a geometric progression. So if you look at this one, his calculated one, so here's the ones he calculates using the last 2.4 times 0.7, he gets 1.7, which is a good match. 1.2 is a perfect match, he gets 0.84, 7 of 1.2, and so on. So he gets, and this is a very good match for a geometric progression. So
and that's something that's still used today?
Yes, it's a tremendous tool. And of course, but what's really interesting is by this time, by 1895, these notebooks that you're looking at, you'll see a lot of these calculations and ratios. He publishes the actual found values, but he does not publish the ratio anymore. And I suspect part of the reason is he found that there were exceptions to the rule and wasn't sure it was that useful. The other thing is, in his original work in 1890, and this is all spelled out in my book, yeah, it's very funny that in 1890 he published all these results, but it doesn't seem like anybody's ever gone back and recalculated and figured out that Dyer used a variety of ways of getting his ratios. Sometimes he looked at the last 2 numbers. Sometimes he pooled all the numbers. He did various things, whatever kind of fit the best. And that's really not clear. In the literature, we think that it's the last 2 that were always used. Though, to his credit, Dyer did a whole bunch of footnotes saying all the exceptions, blah, blah, blah. But I think it just sort of bothered him that it wasn't as good of a fit as he would have wanted. Yet today it's been, I think over the last 30 years, somebody did a survey and 80% of the time it works to be able to predict the stage, what stage you have, or be able to figure out what the earlier instars, later instars are.
Now, I think we've teased folks a lot with these really interesting specimens back here. Would you like to give us an idea of how Dyer's field books tie into what we see here?
Okay.
And how you can understand his collections through his field books. Okay,
so if you look at these labels right here, you'll see Collection H.G. Dyer. So these labels were all on this specimen here. These two labels were on this right here. So
and that
notice
right there is eggs, right?
Yeah, and the eggs, where my finger's pointing, 570 on the label. Well, that little 570 above Collection H.G. Dyer is actually probably the most important label of this whole batch because that ties into
the tiniest one.
Yeah, the tiniest one is actually a Blue Book number. So let's go over to Dyer's 570 entry. And it's right there. And it says Euclea on Cocoloba floridana. So here's another one from his trip to Lake Worth, Florida back in December 27th, 1895. So at that time, he found an elliptical flattened, and of course here I'm showing that I can't always read this stuff either. So it says highest through 5. Highest through 5. So that 5 probably means the 5th segment. Dyer counted his segments counted the head as a segment. So 5 would be head is 1, first thorax 2, second 3, third 4. So 5th is the first abdominal segment on this caterpillar. Anyway, and that's true actually because the Euclea was the highest on 5. So if you look down into the notebook, you see actually very nice little illustrations of the caterpillars in the later stages. And guess what? We have a little blue squiggle.
Which means he published it.
He published it, and here is the publication.
And so this is a copy of his article that was in the Journal of the New York Entomological Society, is that
correct?
Right? Right? Which the Biodiversity Heritage Library actually has a nice run of if you're looking for an online source for it. And I love the illustrations that go along with this article.
So here we have, and what's interesting is these are not in the Blue Book. So these were, I guess, elsewhere he did these drawings, the originals, and they're probably lost forever. But what we have on here is pictures of the larval damage. This is the stage, what we call the first instar, so just hatched out. This is like on its thorax, these tiny little legs. So these are slug caterpillars, and what that means is that the underbelly has evolved into these suckers. And this is what I did my doctorate on, and this is how I got into Dyer. I saw these very articles that he wrote, these incredible tomes, a 20-part series called The New York Slug Caterpillars, which sounds kind of funny, I know, and maybe insulting to New Yorkers, I don't know. But what's amazing about it is that not many people showed these first instars, and they're somewhat diagrammatic. These were done probably with what's called a camera lucida, a little drawing tube where you can actually, as you're drawing, see the image through the microscope, so you're kind of tracing over what you're seeing. So they're very accurate.
And so we just had a question: did he do his own illustrations?
Yes.
And so he did using a camera lucida, but the coloring is fascinating also.
Yeah, his sister Pearl, by this time, hand colored, and only select numbers of them. Yeah, I asked, yeah, I guess Grace is Constantino's gonna, from the BHL, is gonna look into whether all the ones that have been scanned in BHL are the ones that have been colored or not. But anyway, yeah, early on in his first articles, in late 1895, and again, he was the dominant figure in this journal during its early years.
I think you mentioned that he co-wrote.
Yeah, he co-authored with Emily Morton, who was really the expert at the time at finding eggs of these things, which are tiny. And as a matter of fact, again, we'll go back to this. You can't even really see it here, but they're shiny, clear, little flat scale-like eggs, and you can actually watch the caterpillar embryo develop within them. It's an amazing process. So you always know when they're about to hatch.
So the eggs on this pin are actually, but it's not just the eggs themselves. They're
attached to a leaf. Yeah, these you couldn't pull it off a leaf because they're too fragile. So he kept these and kept that blue book number, which ties into that. Now we'll get on to the adult. So notice this other number, 12081. Well, that goes with what we call Dyer's catalog. So Dyer recorded when adults that of the caterpillars he was rearing came out, he would say out, and you'll see this terminology sometimes in the blue books too. He'll say out and then give a date. So that means that's when the moth came out. So oftentimes that's the following year. Out. So these caterpillars, so Dyer, when he was in Florida in December, found caterpillars. They spun a cocoon, and then by June 1896, these two, same day, this is a male, this is a female, they were in the same little rearing cage. They mated, and this female laid these very eggs.
Oh, wow, it's a little moth family.
Right. Now this one came from the same group of caterpillars, but came out at a different date. This one came out on May 27th, 1896. So what I've done over the years with these slug caterpillar moths, or Limacodidae, is through the blue books, I've made new labels. I still keep the original labels because it's very important to document that. We also have barcodes that are in a database of these particular specimens. But now I'm going to show you. So remember the number 12081. It's at the top of this page. It says out from Lake Worth, Florida. And then the date that it's out is May 27th, 1896. But then the most important thing is, look, BB 570 Euclea. So that's what cross-links this information of the adult moth with what was in his notebooks.
And so this is a photocopy of another, I think it's categorized as a field book, but it's Dyer's catalog of Lepidoptera.
Right.
And this is also in the Biodiversity Heritage Library, and we'll send out a tweet with a link to if anybody's interested in doing it. We're on 1,000th entry, but it goes up to 39,000 entries.
Correct.
So
and almost half the entries are from I can't remember, 1903 or 1904, when he went to Caslow, British Columbia. So and it goes through 1913, but most of the records go through 1904, 1905. But throughout this, you'll see blue book entries. So this can also be sometimes a way that you can find more information about what's in the blue book.
So I'm going to back up here so that everyone can see all the things that you are working with to try to tie Dyer's research together. You have the blue books, you have the journal articles, you have copies of his catalog, you have his specimens, and this is some serious detective work here. So this is a lot of material, and amazing to watch it all be pieced together when it comes from so many disparate sources.
Right, and as you can see, the style of the notebooks is a little different. The last in the series is this, and these records are all mosquitoes.
Right, and so Dyer didn't just do moths; he did mosquitoes and also sawflies. And that's interesting that he worked on mosquitoes. I'm curious if any of his mosquito research is applicable today when we think about mosquito-borne or mosquito transferred illnesses like Zika virus and stuff like that.
Yeah, very much. As a matter of fact, at that time, and today too, there's a link between the Smithsonian and mosquito research at the Walter Reed Biosystematics Laboratory out at our Suitland facility. And those researchers are employed by the Army, but work in collaboration with the Smithsonian, much like USDA does. Now, back in Dyer's day, the USDA was in charge of mosquito work. And in 1901, Dyer kind of got bit by the mosquito bug.
Bit by the mosquito bug.
And he started searching for mosquito larvae because at that time, we were looking at the construction of the Panama Canal, and L.O. Howard, in consultation with Walter Reed, was, interested in working out the taxonomy of mosquitoes, because if you don't know what mosquitoes are the ones causing, that are vectoring the diseases, then, it's kind of like with agriculture. If you don't know what caterpillar is causing the damage, you really are not, it makes it more difficult to do the science. And that's because, of course, these are, when I say classified or identified, they're part of, a tree of life, just like we are, and they're related. There's evolution. They're linked to others. So, of course, knowing about one, if you have good taxonomy, you know about others. So what Dyer did is he learned early on that actually the larval stages, kind of like with his caterpillars, he learned with mosquito larvae that they often told more of the story of what species of mosquito the adult was, because there'd be mosquitoes that could not be told apart easily, but if you raised them separately with and associated properly the larvae, which are aquatic, they're in the water, so if you can they had more distinguishing features in the larval mosquitoes than the adult mosquitoes. So we actually named some species based on the larvae, not the adults, which is very unusual to this day. And actually those species have held up.
Wow. And we have, I'm going to go ahead and put the webcam back on the computer so I can stop jiggling everyone around him.
Oh.
And Mark, no, it's my fault. My unsteady webcam hands. And Mark, you and I can have a seat because we have some questions coming in now. One is about the slug illustration. If I went out the yard after this program, would the garden slug look like the drawing?
Well, not exactly. Certainly the drawings are, I'll show you another example. I mean, they tend to, of course, being insects, they're segmented, so you can kind of see little lines separating the segments on the caterpillars. And also there's a certain repetition of design or pattern, each segment has, of course this would be tiny, but they breathe in each. Insects are amazing because they're so decentralized. Every segment basically has a brain, has a breathing tube, has a nerve, like that. And then their circulation is open. So that's, of course, how, I was talking earlier a little bit about blowing caterpillars, and these dry caterpillar specimens. I don't know.
So is that one of the slug caterpillars there?
Yes. This is one of the ones from Florida that we were talking about earlier. I forgot to show it. So, I guess I did show it. Anyway, I don't know. It's probably better to show it this way. So, yeah, there's sort of a pattern. Of course, these have spines. They're slug caterpillars so they're called slugs because of the underbelly, not because of the upper surface. Some of them are more like real slugs because they don't have spines, but they're still more segmented, harder, and also their locomotion. While creeping way like slugs, it's a rolling wave. It's a little bit different. The other thing is this time of year you would never see a slug caterpillar.
Oh, really?
They're late in the season in temperate zones. That's because their whole biology is tied behind feeding on really tough, smooth leaves. So the female moth lays an egg, like Coccoloba in Florida is a classic example. A Coccoloba leaf is a really smooth, tough leaf. So you need a smooth, tough leaf to lay a flat kind of egg, like a lemon-coated egg. And then the caterpillars to stick to a leaf to feed have to have that, that slug surface really helps them. So it's a whole group that's evolved kind of the slug biology.
Interesting. We also had a question about sawflies. The field books, do they contain studies of sawflies or just moths and mosquitoes?
Right, now there's the sawfly notebooks too, and occasionally, kind of like the mosquitoes, there'll be a mixture in a few. So
a few of the field books.
Yeah, so some of the Lepidoptera field books will have a few sawflies thrown in, a few mosquitoes.
And so we only, right now in the transcription center, we only have 5 of Dyer's field books up, but how many field books in total do you think there are?
I think it's probably, certainly low 30s. Some of the 25 that were reported to have aren't really true field books. Some are not by Dyer.
So we do it, I should clarify, there's 5 in the transcription center now, but I think most, if not all, of the field books have been cataloged and scanned and are available through the Smithsonian archives, as well as the Smithsonian Field Book Project and Biodiversity Heritage Library. So the sawflies may not be the focus of the field books that we have that are in the transcription center right now, but they are out there, and we can find you some examples and tweet them out too.
Yeah, one of the things that eventually will be very interesting, and I've started to do it already, is in a database I've pooled all the data from the Lepidoptera field books with those of the sawflies.
Oh, and I see Grace has just helpfully added an answer in the Q&A box. If anybody's interested in seeing his field books on PHL, she's added a link for that.
Oh, great. Of the sawflies.
I don't think they're specific to the sawflies. I think that's just the whole collection.
The whole
Dyer
collection.
field notes.
Okay.
Yeah. So we've talked a lot about Dyer's work, but you can't really talk about Dyer without talking about his bizarre personal life. So what is it that is so fascinating about Dyer, besides his his dedication to mosquitoes and moths and sawflies? What do people really like to hear about him?
Well, of course, they like to hear about the 2 wives and the tunnels.
The 2 wives and the tunnel.
And the story that everybody, at least I heard, many, at least entomologists here, but I guess in Washington, D.C., various times over the years, it's gotten to the newspapers of Dyer being one of the more eccentric of an eccentric group of people. People that are entomologists, I guess. But actually, we're not, I don't think we're that any different from other scientists. Really.
So Dyer had 2 wives, and he was actually dismissed from the USDA as the chief entomologist because of that, right?
Right. He wasn't a chief entomologist, but he was what they called an expert at the time. And yeah, what happened is that Dyer's divorce, what he attempted to make as a quiet divorce from his first wife, and went to the West Coast to do that, to California. Well, he settled in Reno, his future ex-wife in Berkeley, and the divorce took 5 years.
5 years.
But it was in the newspapers. And then what was also in the newspapers was that his second wife, or concurrent wife, was married to Dyer as an imaginary person.
As an imaginary person.
So, well, at least the judges knew that, but she was trying to get a divorce from someone who did not exist because he was Dyer. But he went by the name Wilfred Allen, and they claimed that Wilfred Allen was real. So she could not get a divorce, and it went all the way to the Nevada Supreme Court.
She couldn't get a divorce because he wasn't real.
And the judges were onto them. So this went on also. So both cases were in newspapers at both parts of the country. At that time, it wasn't a national story per se. But what was a national story was his tunnels. And
but back to his wives, he named species after both of them, right?
Yeah, and that's what got me into the whole mess. Is
you stumbled on it by way of the species name?
Well, yes, but also the slug caterpillars turned out were Dyer's favorite, and he named wives after the two wives. But what's interesting is Pam Henson of the History Division at the Archives, who's really the keeper of kind of all entomologists, well, all scientists, the most eccentric of the Smithsonian scientists. She has files on. So when I first came to Washington, people said, you must talk to Pam Henson, because I would mention Dyer merely in a research context, because I said, he really did a lot of work on the group I work on. And so it was always Pam Henson. It was all about Dyer's tunnels. So so I went and met Pam, and I said, oh, by the way, Pam, what was the name of the mistress? And she said, Waleska, which is a very unusual name. Oh, the name of a moth I had just started studying from Costa Rica, Parasa waleska. So I started looking at the literature, and I found out Waleska was Waleska pollock. I went to the library and looked at old city directories and saw that Waleska pollock was part of this family of kindergarten teachers, and that her brother was George Freeman Pollock, who I eventually found was the proprietor of what was then called Stony Man or Skyland, which became Shenandoah National Park. So, and her mother was one of the first to bring kindergarten to this country, Waleska's mother. So it just sort of, so every time I'd find something like this, I come back to Pam, look what I found, look what I found. And so I said, so I said, oh, Pam, what was Dyer's wife's name? And she said, Zella. Yeah, the first wife was also named after one of my moths, or one of my moths was named after his first wife.
But that wasn't until 20 some years later, or?
Yeah, yeah, towards the end of his life, he names it after the first wife, and then I started learning about the divorce, so I knew they had already been divorced like 6 or 7 years. It was frustrating because we didn't have pictures of either one, so when we give lectures on Dyer, we'd show, well, eventually we got one of Waleska. When we met Dyer's son and interviewed him, he had a picture that he gave us. So at least we knew what Waleska looked like eventually.
Before that, you only knew what the moth Waleska looked like, or what the cat.
Before that, but Zella, I didn't know till 5 years ago what she looked like. And I did because even though I've been in California and still as a research associate of the Smithsonian, kept my connections here. I work for California Food and Ag, but out in California, there was this family that I'd met while I was at the Smithsonian. And, you know, over the years, families of Smithsonian curators and staff like to come to the museum to see their specimens. So this family from Southern California and Virginia, there were two brothers, turned out that Dyer's son Otis was their Uncle Oat. They weren't related to him through marriage because Otis didn't have children. But Uncle Oat was like very important to them, and their parting words as they were leaving is that, oh, we have these photos, and there's this Ella person in the photos. And I thought, could that be Zella? But then it took me 10 years of calling one of the brothers, because unfortunately the younger one, who was the real historian, passed away, but the older one moved to California. Eventually the widow, who had the boxes of pictures, ended up in California, and I met them in Pasadena, and lo and behold,
to connect it all together.
Ella was Ella, but there were pictures of Zella. Ella was a different person, but not only that, pictures of Dyer with his children. And also a picture of Dyer with the sandcastle. That looks just like a slug caterpillar.
Oh, he had a thing. So he had the two wives, but also the tunnels. So he had two sets of tunnels in DC. Right. And one was under his residence, or?
Well, one was on the side. That was the one across from the what's now the Hirshhorn. Oh. Kind of in between the Hirshhorn and the Arts and Industry. At that time, the Army Medical Museum was there, where the Hirshhorn is.
And they weren't just like somebody took a shovel and dug the tunnels. They were excavated.
That was the second set that was at his home with Waleska, which he had built for her. But the first set was in DuPont Circle, and that was actually the set that brought the whole thing into the public view, because actually in 1917 was the first collapse, but that was right before the U.S. got into World War I, or right at the time. So it was just a little footnote in the newspapers. It showed that Dyer was the digger, but then 7 years later, they collapse again behind the home, and then that makes the national news. National news.
So do you think that there was any connection between Dyer's entomological work and the tunnels, or it was just a weird hobby?
You know, there's a couple connections. One is, of course, as a researcher, sometimes you are, it sort of almost goes with the territory to be somewhat compulsive, because of course you follow leads and you want answers to your question. And certainly Dyer was an incredible researcher who did, as he said, from small acorns, big trees grow. And that's kind of emblematic of, all his work. You can kind of see the beginning, but then you see this pathway, which is kind of like a tunnel that he keeps following. But then there's another aspect, and that is Dyer, because he came from wealth through his father, who was an incredible inventor. He invented, a telegraph long before Morse, and also communicated with Joseph Henry, who really worked out the electrical part that made the kind of the magnetic part of the eventual Morse telegraph. So between Dyer and Henry and several others, really is what made the telegraph well before Morse. But anyway, and as a matter of fact, his father had a tunnel digging device from 1832 when I first found 1831, and I have no idea. The patent was never filled. There's no. But when I first found it, I thought it was a misprint. I thought, oh, they got the date wrong, and it's H.G. Dyer Jr., not H.G. Dyer Sr. But he applied for a patent, and it was never filled. But anyway, so yeah, I could say maybe there was, could even say the father showed him the blueprint to this, but the connection to his father that he lost when he was 9 years old. But then the other side of it that might be more important is Dyer was into architecture. Because he had the wealth from his father's patents, and actually it wasn't from the tunnel digging device, it was from dyes. He developed, he was a very good chemist and physicist, well, sort of an early physicist in a way, knew about electricity. But he was awarded $400,000 for patents on dyes in Paris, used by the fashion industry. So he came back to the U.S. with this money and invested it in New York real estate. Well heeled, and so when he moved to Washington, he started buying properties from the best architects in Washington. And but then around the same time, he was also digging underground, and and he didn't just dig tunnels. They have archways. They're pretty sophisticated. Use special bricks for them. So one of the kind of biological connections that I've sort of thought about is, caterpillars like Limacodids are very geometric, shape-wise. There's others that are even more so than this. They can be very triangulate, but these kind of have an archway and supporting beams. They're very much like something an architect would think about. And I just wonder if Dyer had an element of that too, and might not have been, he said that he started digging a garden, and you could say convulsive Dyer just kept digging. But I think it's a little more than that. I think also he was in a heap of trouble by then and trying to figure out what to do. It's possible that his mistress had become, with child earlier and lost the child. We don't really know. But digging certainly could have been a way to just also eventually a place to just go down and think or hide. Or also his first wife was deaf, so in the household everybody was shouting, and I think Dyer maybe just had to just get away from all of that. So there's a various.
I saw a question earlier and then it disappeared on me, but somebody had asked if any of the tunnels still exist today.
Well, the three-story ones across from the mall, the National Mall, we can safely say do not exist. They were gone in the 30s, and that whole neighborhood, maybe as well, I guess early 40s, I'm sorry, the house was still there. Yeah, there was even talk during World War II of using those tunnels as a bomb shelter.
Oh, really?
But
were they that stable and
well,
that well built?
I think they were, but I don't think they were big enough. And, you know, they were winding and for whatever reason. So sometime in the 1940s, whenever they destroyed that neighborhood to build, well, I don't know when they built the Department of Transportation exactly. So that's been long gone. But then the DuPont Circle ones probably still exist in some form. The entranceway was certainly blocked off. And there's a picture of it in the book when I visited there in the early, let's say, I guess it was the late 80s. I went over there.
Yeah, and there were some great Washington Post articles about Dyer and his mysterious tunnels, which we can tweet out after.
Right.
Afterwards,
yeah, John
too,
Kelly did a great job with
for
that.
some more information. So I think that we've answered all the questions that are here so far. If anybody else has any, please pop in and ask. Otherwise, you can also tweet us afterwards at SI Libraries using the hashtag #DigIntoDyer. I think that is about all we have for today, but I'd really like to thank Marc for joining us today. Marc is giving a Smithsonian Associates lecture this evening, and you can, for those that are local to DC, there are still tickets available at smithsonianassociates.org. And I'm sure it'll be a fascinating lecture. And of course, Marc, do you want to show your book? It has a new book through Oxford University Press, Moths, Myths, and Mosquitoes, The Eccentric Life of Harrison Dyar. And we would love for folks to jump into the Transcription Center project. We still have a little ways to go on our transcription. Is there anything else a person could look at online? Someone asked. Yes, we will send out some links. You can also go to blog.library.si.edu, where we're having a series of blog posts, guest posted by Marc about Dyar. They started last Friday, and we're sending out lots of tidbits via social media using the hashtag #DigIntoDyer. We'll be sure to send out some of the Washington Post article links, as well as a link to Marc's Associates talk tonight and Marc's book. And we also have some great imagery from Marc that we'll send out to go along with this broadcast. So, oh, thanks for adding the link, Grace. So thanks for everyone for tuning in, and we'll turn to Twitter, and if anybody has any additional questions. And again, if you're in DC, pop in and see Marc this evening. Go to smithsonianassociates.org for tickets there, or take a look for his book. So I also want to thank, before we go, the Transcription Center folks who have helped us put this together, specifically Megan Ferreter, the Smithsonian Institution Archives for helping us get the field books into the Transcription Center, Biodiversity Heritage Library, and the Smithsonian Field Books Project. So thank you all for your help, and we hope you continue, volunteers out there, to dig into Dyar. Thanks. Bye.